So yeah, just to set the stage very quickly, this is a very exciting talk that we have coming up with experts across the genetic editing realm. With no further ado, I just want to very quickly thank again Rebecca, Jimi, Alta, and Julie for joining us today. They each represent such deep expertise across science and medicine, and specifically on genetic editing. So I will let them introduce themselves because I just can't do any justice to that. So yeah, whoever wants to start off, I think. I mean, if Rebecca, if you don't mind, we would like you to start it off.
Hi, everybody. I'm Becca Ahrens-Nicklas. I'm a physician scientist at the Children's Hospital of Philadelphia and University of Pennsylvania. And I have the privilege of taking care of kids with rare diseases. And I run a translational research lab trying to develop new therapies.
Hello, I'm Alta Charo. I formerly was a professor of law and bioethics at the University of Wisconsin on the law and medical school faculties, currently working as a consultant to biotech companies and government agencies. I co-chaired the U.S. National Academy of Sciences report on genome editing regulation and also served on the WHO committee on global issues with genome editing and the International Society for Stem Cell Research activities in this area and the international summits on genome editing.
Hey everyone, my name is Jimi Olaghere. I am fortunate to be one of the first patients to receive a CRISPR-based gene therapy for sickle cell disease. I am a investor in private equity that focuses on small businesses, so please don't send me your pitch decks. I have no idea how to invest in biotech.
Hi, my name is Julie Saba, and I'm a physician scientist and trained as a pediatric oncologist at UCSF. My story is I cloned a gene at the beginning of my career, looking for genes involved in cancer, then found a rare disease caused by mutations in that gene, and am now developing a gene therapy for that rare disease, and started a company with the help of Nucleate. So thank you all for inviting me.
Yeah, thank you. Thank you, Julie. Thank you, everyone. How we're going to do this is, I really want the panelists to have a 30-minute conversation. I'm sure that it's going to be super rich, but just to kick it off, we're going to do one by one sort of rapid fire questions. So I'll start with Rebecca. Our question for you is, you led the first ever single patient personalized CRISPR therapy this year. Probably the most positive news I saw all year this far. Baby KJ, if anybody doesn't know, what did this moment really teach you about the potential and the limits of gene editing as it stands today?
Yeah, thanks for the question. I think it's taught me a lot, both about sort of where we're coming from and where I hope we're going to go in the future. So as I mentioned, I'm a rare disease pediatrician. Or I'm a clinical geneticist by training. Every patient I see in my clinical practice has a rare disease. We know the monogenic cause of every single kid that walks into my clinic. And especially in light of everything that has happened around this first example of a personalized gene editing therapy, every single one of my patients and every one of my colleagues' patients around the country have asked, when is this going to be available for my child? And I think this experience has taught us a lot of things about what is possible, but also what we need to change really in the technology space and in the clinical implementation space to allow this to expand. And so we learned a lot through the experience of trying to develop a personalized gene therapy or gene editing therapy. For those of you who don't know, this is a baby that we care for that has a very severe disorder called a urea cycle disorder that unfortunately it often leads to death in the first week of life. These babies are very, very sick. And my collaborator, Kieran Musunuru at Penn, and I had been working for a number of years to basically gear up to be able to develop a personalized base editing therapy for a patient. So when KJ, this patient, was born last year, we had sort of gotten our pipeline in place and actually tried this for the first time. And we were successfully able to at least manufacture a drug, deliver a medication to him by the time before he was 7 months old. I think there's a lot of things that we learned from this experience. One, it's important to say this was clinical care. This was a single patient expanded access investigational new drug application from the FDA. This was not informal definitions clinical research. Now, obviously, the family was consented. It was treated as a research study, but we can't make broad claims about how well the drug worked or how safe it is even, because it wasn't done in a formal trial setting. And so that's one of the first things we knew going in, but we especially feel now that we need to move forward through formal clinical trials, through platform clinical trials. So we really can evaluate the safety and efficacy of these drugs, because in the rare disease space, reimbursement for drugs like this is very important if we're going to be able to treat the next generation of patients that are born with these devastating diseases. So that's one thing we've knew, but have learned even more so, that we need to move these forward through formal clinical trials so that someday we can have an approved product that can be reimbursed. The other thing that we've learned is that it's possible, right? In a timeline, you really can develop a personalized editing therapy, but you need an infrastructure in place. We built the infrastructure as we went. It was really everyone donating their time and effort, and the stars aligned to make that timeline work. We're not going to be able to rely on fate and luck, essentially, for future children. And so we need to not only build an infrastructure in place for clinical trials, but we need to be able to build an infrastructure that will support a commercial product down the road if we're going to really scale this. And then the third thing we learned is that there's a lot of unmet need that is still out there, right? We worked on a disorder where if you edit the liver, you have a therapeutic benefit, or you may have a therapeutic benefit for a patient. Unfortunately, right now, delivering of editing tools outside of the liver is still very complex and very difficult. And so we learned, which something I think we all knew, but I feel even more acutely now, is that we have to advance our technology to be able to target other organs, because of the thousands of emails and phone calls and requests I have gotten for help since this news broke. So many of them, you'd have to correct other organs like the brain or the heart or the muscle. And so I think we've learned a lot about what we're capable of, but I think we've learned a lot about what we have to do.
Yeah, thank you so much for the 3 lessons. And on the topic of firsts and on infrastructure, I would actually like to jump to Jimi. You were one of the first patients in the world treated with CRISPR for sickle cell disease. Can you speak to that experience, being one of the firsts, and how that has shaped your advocacy and your current work?
It was life-changing, transformative. I went from being bedridden to be able to summit Kilimanjaro a year after getting infused with my own cells. The process was painstaking, giving everything you have to go through to get a genetic therapy, gene editing therapy from the presented a lot of issues. Are you guys hearing me or no?
Now we can. You cut off for maybe 8 seconds. Yeah.
I'm sorry about that. I think it's my noise canceling. But yeah, the collection phase, which for me, like I said, I had to get collected 4 times, which I think we're going to discuss that genetic diversity as we go through this panel. It's been an issue with just the lack of it in clinical trials. And then, of course, the busulfan as well, the 3rd stage, which is another challenge that most patients would have to face if you're going through a clinical trial that was similar to mine, which was ex vivo. And then finally, the infusion, which for me was kind of anticlimactic after going through all of the rigors, all of the headaches to collect enough stem cells, and then they come back with just 2 syringes of clear fluid can give that anticlimactic effect. So that was what the process like. Of course, I felt quite fortunate throughout the whole process to be selected and be able to participate in a clinical trial like this. But what I had come to realize through my advocacy, because the therapy, like I said, quite changed my life, and I wanted to make sure the patient had access to this therapy. So coming forward with my story, my goal was to make sure that everyone was aware. Anyone who wanted a gene therapy, if you have sickle cell disease, if you wanted a gene therapy, you had access to it. But I'd come to realize that the therapy was actually quite inaccessible. For the one I have, which is now known as Casgevy, it costs $2.3 million. And that has been literally the basis of what my advocacy has stemmed from, is the fact that these therapies are approved on the market, they're working, highly efficacious, but patients that need it don't have access to it.
Yeah. Yeah, thank you so much, Jimmy. And, I want to extend that to Julie as well. You're also a pediatric oncologist by training and by practice, and you're currently working on a gene editing technology. And, looking further ahead, how do you see this technology specifically fitting into the treatment landscape for cancers, where, we have an arsenal of chemo, of CAR T, et cetera? What does that timeline look like? What are your main areas of concern, and what really excites you and motivates you every day?
Thank you for the question. So I'm mostly focused on rare diseases now. I'll just briefly address the issues of cancer because although I started out my research career in trying to find new treatments and cures for children with cancer and hematologic diseases, that is not currently my focus. So I guess the three main ways that gene editing or gene therapy can address pediatric or adult oncology problems are to, one, bolster the immune system through CAR T modification genetically. Secondly, to address or attack genes that enable the cancer cell to be immortal, and so induce cell death by knocking out certain genes in the cancer cell. And a third innovative way that cancer cells can be addressed by gene therapy is by enabling them to activate certain pre-cancer or pre-drugs, so that only the cancer cells will turn on the activated state of a lethal molecule, and thereby specifically kill the cancer cell. And there are many other people in the world who are working very, very hard on those approaches, and I think they have great promise. My focus has been on this rare disease that I discovered, and because I'm really the only person in the world who's focused on therapeutics for these children, and I really have come to understand how important it is for each individual rare disease to have its advocate, whether it be the families or an scientist, physician scientist. Somebody needs to be the one to advocate because there are so few of these individuals for every particular disease. I have used small molecules, even a vitamin, to cure a whole handful of patients with this disease. But gene therapy, and in our case, we're not really using gene editing because there are about 50 different mutations so far that we know of that lead to this inactivation of an enzyme through mutation. And there's probably maybe even 100 more because it's a very conserved enzyme, and fiddling with any nucleotide in the gene sequence is likely to inactivate it. And so gene editing would have had to be even ultra personalized to work in this case. There are strategies around that, but to me, gene therapy, which is just taking a working, healthy copy of the broken gene and delivering it through a viral vector, seemed to be the most appropriate strategy. And as Rebecca mentioned, one of the big challenges in gene therapy, whether it be for gene editing or for just straight gene therapy, is targeting. So the natural viral serotypes that usually we use adeno-associated virus, there are other virus vectors that are used, but the one we often use is AAV, and it has a limited tropism, so only certain tissues get targeted. But there's a whole huge, busy activity now with many, many labs and companies developing new capsids that improve the targeting and have been able to reach other difficult to reach organs. And ours in particular, our disease affects the kidney, and which has been particularly difficult to reach with gene therapy. But we have used a genetically engineered capsid that targets the kidney well, opening up new doors for this treatment. I'll just say one other thing, which is that, there are lots of small molecules that people use to try to treat inborn errors of metabolism, but there's nothing better than the original enzyme, because you have the backup of whatever happens before the enzyme is broken, and then you have the absence of products. And there's really no replacement for this kind of disease better than restoring the enzyme activity that's missing. It normalizes the metabolism and has the best chance of a long-term cure.
Yeah, thank you so much, Julie. Yes, I love that last point of, really returning to the basics and respecting that original biology. And I think what we've heard so far is, there's this recurring theme of scale, of this area to reconcile between very rare patients and diseases and the need to build infrastructure. So on that note, just one last question to Alta, and hope that after that, our panelists can engage in a discussion, after which we'll take Q&A. So to Alta, you have advised Congress, the FDA, international agencies for decades, and especially with respect to this problem of scale, what do you think are some of the biggest regulatory challenges that we face right now in making gene editing more safe, ethical, and most importantly, broadly available?
Well, that's a tall order. I think that there are challenges at pretty much every stage of development. They start with the need to diversify the underlying research that gives us insights into the genetic underpinnings of many of these disorders. It's crucial that our databases represent a wide spectrum of humanity, and quite often we are missing large portions of the globe in terms of data collection and analysis, and that limits our ability to understand the origins of the diseases, the biological mechanisms, as well as avenues for targeting something. And the next is that there are just some intrinsic challenges in the manufacturing process. And here I suspect that Becky can speak more than anybody else. But manufacturing for biologics, as opposed to the kind of drugs you think of as like taking a pill, right, is more complicated. It has more sophisticated quality control mechanisms. There are more sophisticated needs in terms of transportation of materials and maintenance of materials. And it's not as easy to create what we might call biosimilars, right? So it's not as easy down the road to create generics, which is another way to drop prices and expand manufacturing opportunities. Then the delivery system, and Jimi talked to this a little bit. I don't know how much people really appreciated what he was saying, but he experienced what he called ex vivo. That is, they took stem cells out of his body through a very onerous process, expensive, painful, in hospital, with a drug regime that had its own side effects, in order to then edit those cells and return them. And that was the infusion that he talked about as being anticlimactic. That ex vivo system makes sense now because you have the greatest ability to access the cells and manipulate them. But ideally, what we want is what, Dr. McCoy and Dr. Crusher did on Star Trek. They would press something into your arm and it would magically go to the right organ and only to that organ. Julie was talking about targeting issues, and would edit the cells in that organ and nobody else. So you need very high levels of specificity there. That is a very big challenge. And it's why she was talking about the other vectors that are being developed, like lipid nanoparticles and other things that might very much preferentially target certain organs, but we are a long way from that. But with that, we would simplify everything in terms of how people can access it. They could go to a local little infusion center and get an injection with maybe some kind of antihistamine to prevent an immune reaction, and then go home, as opposed to having to be in hospital for a month. Then you get into the regulatory process, which is what you had said specifically to focus on. Our regulatory system at the FDA is premised on a model that involves large numbers of people going through clinical trials with a very sophisticated kind of statistical modeling in order to appreciate the adverse event frequencies and severity along with the potential benefits, potentially being able to identify subsets of people or to titrate the dosages properly. None of that applies for rare diseases. And yet we don't really have specifically a regulatory path just for rare diseases. It was under development under the previous administration and the leadership of Peter Marks at the Center for Biologics at the FDA. And right now, I'm simply not sure how far it will continue to advance. In understanding whether the manufacturing standards should be exactly the same, whether the statistical proof requirements should be the same, whether the review timelines should be the same, or whether they need to be specifically adapted to rare diseases. Then with all gene therapies, there's the question of follow-up, because the concept here is that you treat people once, it fixes the problem, and that's it. As opposed to needing a lifetime of taking a pill every day, like we do for statins for high cholesterol, you treat them once and done. But you need to know that it's actually done. And we've already seen some examples in the gene therapy area where some of the effects actually wore off. And because of the nature of the vector, you mentioned AAV vectors, for example, Julie, we weren't able to redose people because they now had a reaction against it. So follow-up is important, but follow-up takes time. And the FDA is looking at like 15-year timelines. Who's doing it? Who's financing it? Who's staying in the same job for 15 years to follow up these patients with some kind of continuity? And all of this leads to the biggest problem in terms of scaling, which is the business model. The pharmaceutical business model is premised on something that has worked incredibly well at bringing innovative drugs to the American market, but it also causes them to be extremely expensive in their early years. Basically, companies invest a gazillion dollars, 90% of the stuff fails, 10% works. The 10% that works gets a special advantage in the market that lets them have basically exclusive marketing opportunities. If it's a novel kind of therapy, they can charge whatever they want. They make a ton of money. They use that money to cover the expenses of the 90% of the stuff that didn't work, as well as to cover the costs of incentivizing the next round of experiments. And it's worked really well, but it is not necessarily consumer friendly. But it's worked okay. And then the generics come in and the prices go down. The trouble here is that you've only got one shot to get your money. You don't get 20 years of 200,000 people taking a drug. You get one shot. And so that's why the price tag, Jimmy, comes in at $2.5 million or $3 million per person. It becomes unaffordable for the person. The insurance companies would make savings over the lifetime of that person, but they often aren't the same insurance company in the lifetime of that person, so they're not incentivized to cover it. And if we ever got to the point where it wasn't rare diseases and it was broadly effective for cancers, Julie, the trouble then is that the public insurers like Medicare and Medicaid would be completely overwhelmed. And we already saw that once with a failed effort by Bluebird in Europe when they tried to introduce a gene therapy to national healthcare systems. They make sense over time, but they don't become viable in the short term because of the high upfront cost that's built into our business model. So we need new financing models that would amortize those costs over time or provide some other way to give a reasonable return on investment and at the same time a reasonable price at every moment in time of the drug's delivery and effectiveness. Sorry, that was a long answer to a short question, but you can't take only one piece of that pathway and solve the problem. They are all interrelated.
Yeah, thank you so much, Alta. So much to cover, across regulation to access to the logistics. At this point, I really want to give the floor to the panelists, but maybe to start us off, I remember from the prep call last week, Jimmy, you talked about the very real logistical challenges that that Alta also referred to. Could you just, for the audience, give us again a glimpse into those very literal logistical challenges that go into receiving a gene therapy?
Absolutely. I think that's one of the things that make the gene therapy, like Alta mentioned, high cost of it. She mentioned I went through an ex vivo gene therapy, which requires stem cells to be quickly transported to a manufacturing center to be edited. If you think about sickle cell disease, it affects the bulk of the patient population are in low resource settings. And to your point, there are no the infrastructure is not built there. So to make this therapy scale in a low resource settings like sub-Saharan Africa, we're going to have to move to in vivo, to make sure that we can move these therapies quickly, get them to patients desperately without having to ship stem cells from one continent to another. Another fantastic option is point of care manufacturing, where we start training people how to collect stem cells in those low resource settings so we can eliminate that high cost of shipping cells across the country and, things getting bad, things getting lost. So that's one of the real factors. Right now, there's actually a bottleneck in terms of gene therapy, the commercially available ones right now here in the US. So I can't imagine how we're going to even make that scale if we are to take care of sickle cell disease and put it on the back burner. So that's a huge issue that I feel in vivo and also point of care manufacturing. And there's been the discussion in regards to if point of care manufacturing is the right thing for sickle cell disease in low resource settings. They want to leave it to the expert, but how do you become an expert at something? It's by actually doing it and getting that 10,000 hours in. So in my opinion, I think it's a good idea to start training scientists in those areas on how to genetically modify stem cells and do it right there in their location, as opposed to waiting to eventually collect the stem cells and then ship them. I think that's going to take a while. Also, I wanted to quickly touch on the point in regards to genetic diversity I was mentioning before, in regards to the lack of data. If you think about it, since the Human Genome Projects in the 90s, I believe about only 2% of the African genome have been sequenced since the 90s. And that speaks to the problem I had during my clinical trial when I had to get collected 4 times. But the principal investigator had no idea why I had to get collected 4 times. It's because we just don't have data. And yeah, the 80% of 18% to 19% are purebred Africans that cover the world population today, yet only 2% of their genome have been sequenced. Also, we have demonstrable proof that all life begins in Africa. So we need to work on fixing that lack of genetic diversity, because it's severely going to help and improve the clinical trial outcomes that we have right now. So like you said, there's so many issues that we have to fix in order to get this gene therapy, for lack of a better term, ready for prime time.
Wow, yes. I mean, where do we even start? So many things to fix. Rebecca, Julie, would love your input on this. There's no single answer, I'm sure, but what can we do in the immediate future right now? What are we not doing right now that we have to be doing?
I think there's a lot of good work being done, but I think there's a lot of blind spots where we need to expand our efforts. And Jimmy spoke to a great one, which is lack of information about diverse genetic populations, but also lack of access to genetic testing, right? You can't treat a disease that you don't diagnose. And so even within the United States, there's a huge discrepancies amongst who has access to rapid genetic testing for diagnostic purposes. And that is even more apparent outside of the United States. And unless you can diagnose a patient, you can't offer a targeted therapy, whether it be gene editing, gene replacement, whatever it is. You have to actually know the disease. And so increasing access to rapid genetic diagnosis is really, really important as a first step, because you can't give a targeted genetic therapy unless you know what you're treating.
I'll just echo that and just tell you a little story that one of the patient's mothers that I care for, she had genetic testing. She was a hospital administrator, and she was knowledgeable, and she was financially able, and she had genetic testing. But at the time that she had her genetic testing done, this disease that I discovered was not yet discovered. And she didn't go back and get a second genetic test when she was pregnant, or just before getting pregnant with a child that turned out to have this disease. So she's now out in the world advocating for people getting repeat genetic testing, because it should be done frequently, because we're constantly finding new diseases that are caused by genetic problems. I just want to say that I love what Jimmy was talking about in terms of expanding the diversity of our genetic knowledge, and also access to these unique therapies. I went to a gene therapy meeting a couple of years ago and attended one of their international sessions. And it seems like there are activities going on in Africa to make this more portable. And I really hope that that continues. And then the last thing I just wanted to say, echoing what Alta was talking about, one of the other problems in rare disease that I think hasn't necessarily been touched upon is not just the genetic diversity of the patients, but also the phenotypic diversity. So patients with rare diseases, they have a lot of different ways that their disease presents. Even children in the same family with the same genetic mutation, they have different modifying mutations. They have different genetic backgrounds from their mother and father, and they just inherit a different collection of genes, and their disease presents very differently. But then even between families, it's even more problematic. So one disease may present in one child with a kidney problem and an endocrine problem in another child. That makes doing clinical trials very difficult because you cannot have one endpoint, which is what every clinical trial wants, is a very clear, simple, single endpoint or major endpoint. And the FDA, if we had our wishes, would accept a couple of other strategies to addressing this, such as having clinical endpoint scoring systems that can unify people with different presentations. So like a multi-domain responder index, but there are others that just score the severity of disease. Is it the same? Is it better? Is it worse? And each individual patient can have their own set of symptoms that they are scoring. And the other is a very important one, which is biomarkers. And there are blood biomarkers, and many of these diseases are metabolic diseases. And the metabolism is the thing that unifies all of the patients, the problem of metabolism. So having biomarkers that can be measured in the blood or urine that can be acceptable primary endpoints is another policy issue that we continue to fight for. Thank you.
I'm going to just add very briefly something that also was suggested in one of the chat comments. We have an opportunity to really focus on both neonatal screening and prenatal screening, as well as potentially carrier screening prior to pregnancy. Because in many diseases, the earlier that you can diagnose, the earlier you can begin to think about options for treatment. And that includes prenatal options, because there's increasing interest at the research level in doing genome editing on fetuses in utero, not heritable editing, just curative somatic editing, because some disorders actually begin to cause damage even before a child is born. But to do that, you really need to expand this access to genetic testing, so you have time to ramp up all of your thinking about what we might need to do, how to manufacture it, how to deliver it. And right now, particularly with neonatal screening, we've got 50 different states with many variations in what they screen for. And with prenatal, we have a very complicated picture politically on reproductive rights that is interfering with the management of pregnancy and is potentially interfering with our ability to move forward even with prenatal therapies that everybody could support. So at a state level, one can certainly lobby for expanding access to these kinds of genetic tests at both prenatal and neonatal levels to at least get us ready to help people, even if we're not there yet.
Yeah, thank you, Alta. We have about 10 minutes left. Just want to really expound on that topic of early testing and access. I think I actually have a sort of complementary viewpoint to offer. I used to work in an inborn errors of immunity lab focusing on infectious diseases, and this is the go-to lab for that. And so we had access to about 30,000 patient sequences in our databases, and overwhelmingly this was, I would say 70% Caucasian patients, Caucasian sequences, and mostly from North America and Europe, because these were sort of after the fact testing that happened when someone presented with a very severe case of an infectious disease. Then they would test, and then we would have the data. And so you can imagine, like, in a proactive world where we're actively doing this testing, we can catch these things so much earlier. And yeah, sorry, Rebecca.
I don't mean to interrupt. Go ahead.
No, I wasn't really going to make any point.
I agree with you, but I think it also harkens back to something that Jimi said, right? So we do, and there's some questions about this in the chat about both follow-up testing after exome sequencing or genome sequencing. So yes, you can reanalyze data as we learn more information after widespread genetic testing. And then someone asked about using whole genome sequencing, which is great, but right now we don't clinically really know how to interpret very much beyond the exome. So even it's great to have that data stored down the line as technology advances. But I think it's really important for us to also realize, to the point you were making, if we are going to be interpreting testing from, at a population level, let's say through newborn screening or however we're going to think about it, we have to know what normal variation is we really have to understand that from all backgrounds and all ancestral groups. As the one, our group gets those phone calls about the newborn screens that are abnormal in the middle of the night, and it's a very terrifying process for families. And if we're going to do genome first newborn screening, which I know some folks are thinking about, we really have to be able to understand what is normal variation and what is disease before at a population level, we're just like, we're putting that process onto families, because coming in with uncertainty because of an abnormal genetic testing result from a newborn screening initiative could cause a lot of anxiety, and it might just be normal variation in their ancestral group. So while I think it's really important to try to understand at a population level these diagnoses, we have to realize that we haven't done a good enough job of understanding normal variation at this point.
Yeah, that's such a critical point, that population level understanding. That's what, this lab and every researcher who does inborn errors of immunity or metabolism really strives for. And I think on that topic, one last question to Jimmy, as we sort of transition from this panel in the next 10 minutes to the panel on equity in science and medicine, what is your sort of call to action, or what are you working on right now? Not only with respect to sequencing the African genome, which we barely have done, but more broadly.
I think three main things, and what I'm working on, what I would like to see with genetic therapies for rare diseases. The first one is the patient voice. That's becoming increasingly important. Take Julie's point with clinical trial endpoints, for example. For sickle cell disease, the endpoint for the clinical trial was pain, but that sickle cell disease is so much more than pain. Through engaging with patients, we came to realize that some patients are comfortable tolerating more pain if they weren't as tired. So we wouldn't have come to that conclusion or found that out if we weren't engaging with patients. So when we're in that process of drug development, it's very, very important in the beginning to have the patients develop that drug at the get-go, not as an afterthought or just checking off a box. Another thing that I'd like to see is improved healthcare system. I think Alta touched on this in the beginning. I think for gene therapies in particular, new technologies deserve new business models. There's no way that we're going to, for all the rare genetic diseases out there that gene therapy or gene editing could potentially use to alleviate, there's no way we can cure all these patients with the broken healthcare system that we have that's strictly based on value right now. We need other outcome-based or different payment systems in order to make sure that patients can truly access this therapy. And then finally, I think one of the most important things for me is gene therapy is so often looked at as a one-time therapy. It's almost like a silver bullet. You get it and one and done. And I think we need to start walking away from that description of gene therapy, because what I've experienced and what I've come to realize is gene therapy, although it's a one-time treatment, you're going to need care for the rest of your life. Even if it's not healthcare, you're going to need mental health care, social support, all of these things that, particularly if you receive a gene therapy late in life, that disease is basically who you are. It's the foundation of who you are. And when that foundation is gone, there's a lot of adjusting that needs to take place that, unfortunately, because it's a clinical trial, these drug developers are creating gene therapy, infusing patients, and, go live your life. And I want us to stop doing that and making sure we take a holistic approach to gene therapy, that once a patient is infused, that's not the finish line. That's where life actually gets harder. And we need to be there to support that throughout life post-transplant.
Thank you, Jimmy. Alta, you have something to add, or should I? Okay, so yeah, we have a few minutes left. I see there's an audience question. I'm going to actually repackage just a bit. So the question was whether you encountered new diseases that nobody has ever seen. I will repackage this slightly to be, what keeps you on your toes? What gets you out of bed? Because we talked about all of these grand challenges that we have to surpass, many of them interlocking, systematic, many things out of our control. What keeps you going? Maybe a rapid fire, one 1 point each. I think that'll be a really good way for us to round off this panel.
I can just quickly say that, yeah, we have talked about a lot of both technical and social and regulatory challenges, and they are all there, and they are difficult, but I really believe we will overcome them. And I just want to hopefully end us on a positive note, because, the story of what happened with CAR T therapy is very instructive to me all the time when I'm feeling down. Carl June couldn't get any grants for his CAR T therapy, his immunotherapy at the beginning. Nobody paid any attention. There were obstacles, technical obstacles that needed to be overcome. And then at some point, he plowed through them, and everything kind of dissolved away. And this is now the forefront of cancer therapy. So these challenges are there, but we cannot run away from them. And companies and economics and all of the problems that we face right now with gene therapy and gene editing are big challenges, but we must pursue the answers, and they will come.
Yeah, thank you, Julie. Any other last points, maybe points for hope, maybe something we can look forward to?
So one thing I would like to say is that current events notwithstanding, we saw a tremendous surge of interest in equitable access to therapies when we saw what happened during the pandemic, and when we saw how much things like the sickle cell treatments are not getting to the people who need them, particularly people in, let's say, West Africa. And I have a lot of optimism that no matter what happens in the short term, in the long term, this is an emphasis we're going to continue to have. And I think this panel has demonstrated it, as well as the questions in the Q&A. That is something that makes me optimistic, because that is what makes me think that in the future, we are going to see the whole world benefit from these technologies and not just a few.
Yeah, I want to leave on a positive note as well. I'm a big fan of gene therapy because I received one, and these headwinds that we're facing, it's actually par for the course. An analogy I've seen is like similar to the iPhone when it was first created, how expensive and bulky it was, and now they're easily accessible. I think the same thing is going to happen to gene therapies. Eventually, the technology is going to proliferate. We're going to keep patients in low resource settings alive long enough through newborn screening and hydroxyurea for them to eventually receive this therapy. So this is actually what's supposed to happen. This is the course of things to happen when new technology is introduced to the market. There's this saying I like to go with is pioneers get slaughtered, settlers prosper. So eventually, of course, because this is so new, we're going to see all the bad things. That's the point of going through a clinical trial, in order to come out on the other side and figure out the things that we need to iron out. So this is here to stay. It's a fantastic option to have for all rare diseases. And I'm like everyone here that, it's still lots of positive, lots of hope.
Yeah, just to piggyback off that, I agree entirely. And you asked what keeps you going. For me, it's the patients, right? It's an amazing time, as challenging as a time it is absolutely amazing time to be a physician, a scientist in this country. And I meet families. Like I said, I meet a lot of families through newborn screening. So these are babies that are 5, 6, 7 days old. And the families ask, well, what's the therapy that's available to my child now? And I'll tell them if there is and what we do. But so many conversations now end with, but I have no idea what's going to be available for your child in 3, 5, 10 years, because the pace of change and the pace of advancements that are happening in the rare disease space is overwhelming, even with all the headwinds. And so I think that it is a time of great hope, even though we all have to be realistic about the challenges. I think in reality, we're going to be sitting in a very different space in the rare disease community 5, 10 years from now.
Thank you so much, Becca, Julie, Jimmy, and Alta. This was really a true pleasure for us to gather the 4 of you, representing, really fearless leaders across the domain of gene therapy. This is, going to be the panel that I refer anybody to. If they ask me about gene therapy, I'm going to be I'm going to say, go watch this. So again, thank you so much. It was a pleasure to host you.